Howdy, Aztecs. How's it going? This is Chapter 14, Epidemiology, Part 2. So here we go. A wild ride. Infectious disease. Makes me think of the coronavirus is quite infectious. So infection then is the invasion of a pathogen. Well, what is it invading? Your body. Yes, your body. That's infection. Now you can be infected, but you might not get the disease. So just because you're infected with something doesn't mean that you definitely will get the disease. But it does mean that the pathogen has invaded your body. And we've said this before, this type of pathogen, a strict pathogen, will always cause infectious diseases. Examples of strict pathogens include the virus that causes influenza, the bacteria that causes tuberculosis, and the rabies virus. They're all strict pathogenic microbes. So like I said, you could be infected with a pathogen, so it's invaded your body. If the pathogen multiplies sufficiently to adversely affect your body, that happens, then you have the disease. But it is possible to be infected and not have the disease. Like this guy, the guy who urinated in the water pipes in England, and he was infected with Salmonella enterica, caused 43 people to die. So think about that next time you go to the bathroom wherever you want to. So some examples. If you're a nurse, you can be contaminated with Staph aureus, and it gains access to the inside of your body, so into the blood. And once a microbe is in your blood, it can go to any sterile organ or tissue that it wants to. Gained access to inside the body, that's an infection. And then you can even break down infections, like a systemic infection. It would be in your bloodstream. You could have a local infection, like a boil. If you could be addicted to drugs, you share needles, you get contaminated. The needles are contaminated with HIV, but you may not, you as the drug addict who were exposed to the contaminated needles, may not see any changes in your body for years. So you're infected, but you're not diseased at that time. All right, manifestations of disease. What does disease look like? So there's signs, symptoms, and syndromes. So a symptom, when you have a symptom, that's a subjective characteristic of a disease. So it's felt by the patient alone. So pain is subjective. Even though I know they try to, I think they give you cards like rate your pain today from 1 to 10. But it's still a symptom because it's still subjective and it's just felt by the patient alone. The doctor can't or nurse cannot objectively measure it. Signs, however, are objective manifestations of the disease, and they can be observed and they can be measured. So for example, a doctor can measure the swelling. You can look at a rash or redness, or you can take your thermometer and see that someone has a fever. And then a syndrome is a group of symptoms and signs that are characterized by a particular disease. So for example, AIDS and SARS are characterized by syndromes. And then this table just shows you some additional symptoms and signs. The study of disease is called etiology. So now there's a whole bunch of etiology. There's etymology, which is the study of the origins of words. There's E-N-T, entomology, it's the study of insects. There's epistemology, that's the study of how you know things. And then there's this word, etiology. So whenever I use one of these words that sound alike, I have to just pause for a minute and make sure I'm choosing the right word. Etiology is the study of the causes of disease. Not every disease results from an infection, as you know. You could have a hereditary disease that's genetically transmitted. Congenital diseases are present at birth, so they could be hereditary. They could be environmental, or they could be an infectious disease. So if somebody has a congenital disease, I think a lot of people just automatically think that's inherited, but it could be, but it could also be environmental or infectious. For infectious diseases, microbiologists use Koch's postulates. We've talked about those in the past, but I think I have another. The next slide talks about Koch's postulates again. So let's look at this. Is kind of cool. Categories of diseases. Hereditary diseases. So the description is caused by errors in the genetic code. You got that from your parents. Examples would be sickle cell anemia, diabetes mellitus, and Down syndrome. Look at all these other categories of diseases. Could be degenerative, that's when just results from aging. Nutritional, if you don't eat the right stuff. Endocrine categories of diseases, hormones. Mental categories of diseases, emotional, psychosomatic. Immunological categories of diseases. So for example, autoimmune diseases. Neoplastic category, infectious. Iatrogenic, this is interesting. Iatrogenic cause of disease is caused by medical treatment or a medical procedure. Idiopathic category means you don't know the cause of the disease that you have. And nosocomial is a disease acquired in a healthcare setting. And I don't think they use the term nosocomial anymore. They use hospital-acquired disease or syndrome. Hospital-acquired. I'm not sure, but nosocomial is the old term. Just to remind you of Koch's postulates. So he was trying to figure out what the cause of a disease is, and he went through this procedure. So in this cartoon, it shows these diseased rats, and he took samples. It looks like he must have cut them open here. I don't know, I'm not sure, but he found them in their GI tract or something, and he grew the microbes, in this case bacteria, on these plates, and he saw there was this colony, so he chose that as the possible cause of disease. He streaked a plate, so you can see how it's streaked, and so he isolated the colonies here so he could count them, and they all look the same from this one colony. And then he could scrape a little of this off of one colony. You could scrape some microbes off of one of these colonies and put it in some fluid, inject it into another rat. The rat dies, and the scientist is able to extract the same microbe from this diseased animal. Okay, so this is kind of cool, the virulence factors of infectious agents. So how is it that infectious agents make us sick exactly? So there's a couple terms. Pathogenicity, that's the ability of an organism to cause disease. So you could be a very highly pathogenic infectious agent. That means you are really likely to cause a disease in your host. That's pathogenicity. Virulence. Virulence is just the degree of pathogenicity. So if you are an organism and you are highly virulent, that means you have a high degree of pathogenicity. That means you are 100% or close to 100% able to cause the disease in your host. So this chart shows at the top more virulent and at the bottom less virulent. And so at the top, very virulent microbe is Francisella turulensis. So it looks like a bacillus. Anyway, it causes rabbit fever. Don't really know much about rabbit fever, but check out this guy. This looks like a Vibrio, right? He's a little curved bacteria. Yersinia pestis. Everyone's heard of the plague. Remember the black, well, you don't remember it, but from history, you've probably heard about the plague. I think, I don't know, the 12th and 13th and 14th centuries, I think, all had plagues. And then if you've heard of whooping cough, that's less virulent than the plague. And infections of burns, Pseudomonas aeruginosa, infects burn victims. That's less virulent than whooping cough. And then Clostridium difficile, or C. diff, antibiotic-induced colitis, is on the lesser virulent. So you could have C. diff and it could cause antibiotic-induced colitis, but it's less virulent than if you got Yersinia pestis. And then Candida albicans causes vaginitis and thrush, and lactobacilli and diphtheroids are all less virulent. The definition of pathogenic itself is disease-causing, and then virulence is the degree of damage that that pathogenic microbe does. Okay, check out this cartoon. So here's a Streptococcus pneumoniae. It's got this sweet capsule around it to protect it, and it's kind of sticking its tongue out here, sticking its tongue out to a macrophage, or it could be a neutrophil trying to phagocytose this Streptococcus pneumoniae, but sorry, macrophage, it's got a capsule on it. You can't quite sink your teeth into it. So virulent pathogens, that's like Streptococcus pneumoniae, cause a pathology to the host, that's you, and they have one or more of the following virulence factors that non-virulent microbes don't have. So non-virulent microbes won't cause disease. Virulent microbes or virulent pathogens will cause disease. So virulence factors include the ability of your pathogen of interest to adhere to a host cell. So that means, I mean, if you are a microbe and you're trying to get into a cell in our body, you have to be able to stick to it. That's called adherence. If you are a pathogenic microbe, you might adhere to one of our cells, but can you enter the cell? Can you invade the cell? And then once you're inside the cell, can you get access to the host nutrients and the machinery of the host? Like say you're a virus and you want to make more of your viral proteins. Yeah, you get inside the cell, but then can you get access to the protein building machinery of the host cell? And if you are a virulence factor of a virulent pathogen, you have to be able to escape or evade detection or removal by the host immune system. And that's what this cartoon is. So this guy, Streptococcus pneumoniae, is evading detection or escaping detection because here's the macrophage going, what is this thing? I'm trying to sink my pseudopodia into it, but it's not working. More virulence factors include your virulent pathogen could secrete an extracellular enzyme that could help the pathogen do its thing. The virulent pathogen could secrete a virulence factor called a toxin, and the virulent pathogen could secrete a virulence factor that was an anti-phagocytic factor. So that means, the macrophages and neutrophils try to phagocytize the pathogen. If you are a pathogen and you secrete an anti-phagocytic virulence factor, then the phagocyte can't eat you. So human 0, virulent pathogens 1. And then I have over here, it says bacterial capsules, like in this cartoon of the capsule of Streptococcus pneumoniae. Bacterial capsules can mimic host connective tissue. So a macrophage or neutrophil might try to detect Streptococcus pneumoniae in its capsule, but the macrophage or neutrophil would detect it as normal connective tissue, and it wouldn't destroy the bacteria. Okay, we talked about this. Adhesions or adherence, it's a virulence factor. It allows you as the virulent pathogen to stick to your host of interest. So after entering the body, pathogens must adhere to cells to be successful. So they need to get inside cells. So they could get in your body, they could be in your bloodstream, but they have to get into the cells in order to be successful. And we've talked about this with viruses, but in this cartoon, it shows a microbe, so it's probably a bacillus, and it has these things sticking out of it here. These are adhesion proteins, and they attach to host cell receptors. So adhesion proteins are on the microbe, the receptors are on the host cells, and if they match up, boom, the microbe has successfully attached to the host cell. So if you are a virulent pathogen and you colonize the human body, then that could potentially lead to infection. Pathogens, virulent pathogens, and they're typically require adherence factors, which are a type of virulence factor. They require those adherence factors to colonize the human body. And adherence factors include these, well, on this microbe, in this cartoon, the adhesion protein or the adhesion factor is called a ligand, which is just a really general term for a protein that binds to a receptor. Adherence factors also would include spikes, like we've seen on coronavirus. The virus has spikes. Pili could be an adherence factor. And fimbriae could also allow a bacteria to stick to a host cell. Okay, in this cartoon, it shows human immunodeficiency virus, and its spikes. In this cartoon, it shows GP120. That's a spike on a virus. And here's the RNA, nucleic acid inside the virus. And then there's, anyway, and then there's the CD4 lymphocyte. So that's a T helper cell. Remember that. CD4 cells are also called T helper cells. And this cell has a receptor, so it's called a CD4 receptor. And you can see here that the viral spike and the receptor, the viral spike on the virus and the CD4 receptor on the helper T cell match. Now the virus has attached. Okay, so back to our cartoon. So here's our micro. We could, in this case, let's just come down here and look at the check mark. A strain of Neisseria gonorrhea, so obviously that causes gonorrhea, has a mutation in its genes which caused the microbe to lose the ability to produce fimbriae, therefore making, and which was an attachment factor for Neisseria gonorrhea. Now so it's got a lot of proteins in it. So you mutate the DNA in Neisseria gonorrhea. Now the mutated DNA can't make a functional fimbriae. Now Neisseria gonorrhea becomes less virulent because they can't attach to the cells of interest. So the pathogen, Neisseria gonorrhea, lost the ability to adhere to cells of the body. So that's good if we don't want that infection. Okay. And it just says here, for example, Neisseria gonorrhea has adhesins that are on fimbriae. So they've got adhesion proteins on the fimbriae that allow them to adhere to epithelial cells lining the urethra and vagina. Okay, so here's another cartoon, adhesins. So these little purple things are the adhesion molecules in the bacterial cell wall. And then you can see how this is cut out. The adhesion receptor is cut out on the host cell membrane, and they match. So you can see over here they're matching. So things like fimbriae, the hooks on a flagella, pili, spikes on viruses, they all allow adherence of the microbe to the host tissue. Some microbes have more than one type of adhesion, and some, like malaria, change their adhesions over time, and that helps them to evade the body's immune system in the evolutionary arms race between us and microbes. If you lose your adhesions, you often lose your pathogenicity. You become avirulent. That means you're not virulent anymore. If you put an A in front of a word, it means that word. It means the opposite of that word. Some pathogens don't attach to host cells, but they attach to each other and then form this web of bacteria, and this is a biofilm. So this is dental plaque. Okay, more virulence factors. The pathogen may secrete an extracellular fluid enzyme that dissolves structures in the body, the host body, and allows those pathogens to invade further. So let's take a look at this. Hyaluronidase and collagenase. So here's your epithelial cells. Here's the collagen layer underneath your epithelial cells. Here's bacteria trying to get in. So they're at the top there, the epithelial surface here. The bacteria secrete hyaluronidase, and hyaluronidase digests the hyaluronic acid that's between these cells. Now check it out. Oh, sorry, the hyaluronidase now is this green molecule, and then these purple things are bacteria cells. So bacteria secrete hyaluronidase, digest hyaluronic acid. It's between these cells. But now, check it out, the bacteria get to this structure. So they digested hyaluronic acid, but now they have to digest or break through collagen. So they secrete an enzyme called collagenase. Wow, 2 for 1. Way to go, bacteria. Now the bacteria can get into the deep tissue, and guess what's in the deep tissue? Capillaries. And then if the endothelial cells of the capillaries are separated a little bit, maybe in some inflammation, bacteria can get into your bloodstream. That's bad. Bacteremia. Okay. So these are extracellular enzymes. It means the bacteria secrete them. The bacteria secrete extracellular enzymes out here in the extracellular fluid. Okay, a few more extracellular enzymes would include coagulase. So you can tell A, so you know it's an enzyme. Coagulase actually coagulates or clots the blood, and that allows them to hide, the bacteria to hide within a clot. So, wow, crafty little devils. So here's the bacterium, here's the virulence factor, the extracellular enzyme called coagulase, causes a blood clot to form, and then look what happens, the bacteria hide inside the clot. Then, I guess, when they, at some point in time, they dissolve the clot with a kinase, an enzyme called a kinase, and now they can get into the blood. Coagulase enzyme can be detected in order to identify some strains of Staphylococcus aureus. So that'd be like the biochemical test that we do in the lab. You could do a coagulase test to see if you have Staph aureus. Kinases, there's a couple they talk about in this cartoon, staphylokinase and streptokinase. So Staphylococcus, Streptococcus, staphylokinase and streptokinase dissolves the blood clot and allows invasion of bacteria into the damaged tissue. More extra, so this is both of the cartoons put together, extracellular enzymes, and talk about some examples. Among the virulence factors produced by Staphylococcus aureus, they have hemotoxin. So hemo is blood and a toxin is toxin. We talked about this one, coagulase from Staph aureus, allows them to hide inside a blood clot. Hyaluronidase breaks down hyaluronic acid, allows them to get between the cells to get into deeper tissue. And then Staphylococcus aureus also produces an enterotoxin. So anytime you see the word entero, that means your GI tract. And then we talked about what hyaluronidase does. It says here it digests host connective tissue, but it's the connective tissue specifically made of hyaluronic acid. And then we talked about coagulase as a virulence factor. Okay. Exotoxins. We're going to talk about exotoxins, and then we'll talk about endotoxins. So exotoxins are virulence factors, and they're secreted by microbes. So in this cartoon, this purple bacterium is secreting this exotoxin, and the exotoxins can harm the host tissue, or the exotoxins can trigger the host immune system that will cause damage, so for example, inflammation. So in this cartoon, the exotoxin is killing the host cells. And if they destroy the host cells and they get down here to the lower tissue, guess what's down there? Capillaries. Bacteria gets the capillaries, can get into the bloodstream, can find a sterile pancreas or liver to go grow on. They just won that round. So toxemia is toxins in the blood. Then septicemia, so these emias, toxemia, toxins in the blood. Septicemia is an infection in the blood. And septicemia would imply that there are actively dividing bacteria in your blood. Viremia, so emia is blood, is the presence of viruses in your blood. So all the emias, you have to know those. And then exotoxins include subcategories cytotoxin, neurotoxins, enterotoxins. So obviously, cyto is cell, so it's a toxin that kills the cell. For example, Corynebacterium diphtheriae. Neurotoxins, nerve, obviously, nerve damage, Clostridium makes neurotoxins. Entero, I told you, is the gut that affects cells, or the enterotoxins are toxic to cells lining the GI. Your body can protect itself from exotoxins by making antitoxins. And an antitoxin is just an antibody made against a toxin. So they call it an antitoxin. And those antibodies can neutralize the toxins so the toxins can't kill you. Examples are toxins that can cause these diseases: gangrene, botulism, tetanus, food poison. This is interesting. Tetanus toxin causes rigid paralysis. So rigid, you're paralyzed, your muscles are stiff, they're rigid. But botulinum toxin produces a flaccid paralysis. So both tetanus and botulinum toxins cause a paralysis, but one type of paralysis in your body would be where you're rigid, and the other type would be where you're flaccid. You can't, you're weak, and it's like a flaccid paralysis is opposite of a rigid paralysis. Gas gangrene, so check out, oh, that's so nasty. Blackened dead tissue and the bubbling of bacterial gas waste in gas gangrene. So it's a condition where metabolic waste produced by bacteria contribute to the pathology. So this gangrenous toe. Cause Clostridium species, it's usually Clostridium perfringens. And you can, Clostridia perfringens as an endospore can get into dead tissue, such as a gunshot wound. So I don't know if anybody likes all those historical Civil War movies, but they had gangrene all over the place from the gunshot wounds. Okay. So this is a table that I made of the bacterial exotoxin and then the target pathology. So bacteria can secrete an exotoxin called hemotoxin, and that toxin will target your red blood cells. Some bacteria make enterotoxins. Those enterotoxins target the epithelial cells of your GI tract. Some bacterial exotoxins would include a cytotoxin, and an example of the target would be the epithelial cells of the skin or your mucous membranes. And then there's a supertoxin or super antigen will hyperstimulate T cells. Now, if you've hyperstimulated your T cells and they're cytotoxic T cells, they could go destroy your own cells that aren't actually infected. All right, those are exotoxins. Now let's look at the virulence factor called an endotoxin. And we've already talked about this a little bit. In this cartoon, it shows, here's phagocyte. This big guy's phagocyte. It has phagocytized a gram-negative bacteria. And if you remember, gram-negative bacteria have that outer membrane, and in the outer membrane of the gram-negative bacteria is lipopolysaccharide, or LPS, which is very virulent. It's a highly rated virulence factor. So then when the phagocyte exocytoses the parts of the bacteria that it chopped up, it also releases that LPS, or the endotoxin that was in the outer membrane of the gram-negative bacteria. And endotoxin called lipid A, or lipid A is actually a component of LPS. Now, sure, the bacteria are dead, but because you, the phagocyte, chopped them up, you chopped up their outer membrane that was full of LPS, and now the LPS as a toxin is released and can cause lots of different things in your body, like fever, inflammation, diarrhea, shock, and blood coagulation. So an endotoxin in general is a membrane-bound toxin which has systemic effects, and it's heat stable. And then I gave you the example of LPS. Endotoxins are released when gram-negative bacteria divide or die or are digested by phagocytes, as we talked about in the cartoon. They're not secreted. Endotoxins are not secreted. E. coli can produce endotoxins. LPS, the endotoxin from gram-negative bacteria, causes all these things: inflammation, diarrhea, hemorrhaging, and blood coagulation. It's potentially life-threatening. Yeah, and then this is just bringing home more stuff about LPS. And I told you they're found in the outer membrane of gram-negative bacteria. They produce a fever, so LPS is pyrogenic, because it can make you have a fever. It could lead to shock if it gets into the bloodstream and causes really super low blood pressure, because the endothelial cells here lining the blood vessel separate and fluid leaks out, blood volume goes down, blood pressure goes down, and now you have shock. And if you go into endotoxic shock, you'll get a fever, hypotension, I just told you that, low blood pressure, and really weak muscles. Okay, so another table I made of bacterial toxins. So the bacterial toxin of interest is botulinum. And botulinum toxin inhibits the release of acetylcholine at the neuromuscular junction. Maybe you remember that from 202. It inhibits muscle contraction. A bacteria release the toxin, the cholera toxin, and this, you don't have to know this, but it activates adenylate cyclase, resulting in higher levels of cyclic AMP, and this leads through a bunch of cool biochemistry and cell biology to super diarrhea, and you lose a lot of fluids and you die. Bacterial toxin of interest, diphtheria toxin. When that gets into the host cell, it inhibits protein synthesis. If you can't do protein synthesis, you can't survive as a cell, so the epithelial cells die. A bacterial toxin called tetanus toxin inhibits muscle relaxation, so that would, you can't relax, that would mean you're rigid. More virulence factors of infectious agents, anti-phagocytic factors. We kind of talked about that. This capsule around the bacterium is an anti-phagocytic factor. So now the phagocyte can't eat you. So your body in general is defending itself by getting rid of pathogens using phagocytosis to eat the pathogens with Toll-like receptors on the phagocytes and PAMPs on the bacteria. But if the bacteria have mutated or evolved such that they can evade phagocytosis because now they evolved a capsule, well, they just won that war. The waxy cell wall components of mycobacteria, that's a virulence factor, an anti-phagocytic factor. You can evade phagocytosis if you are a pathogen by using anti-phagocytic chemicals. An example would be M protein is a anti-phagocytic chemical. It's produced by Streptococcus pyogenes. Broadly classified, it is a virulence factor, and what it does, M protein prevents C3b oxidation, and all that means is the complement proteins, specifically C3b, complement C3b, can coat a bacteria. So you just coat it with these complement proteins, and then that makes, you coat a bacteria with the complement proteins, that makes the bacteria look even more delicious to phagocytes. So that opsonization is just coating. It's a process of coating the bacteria with C3b. And M protein produced by Streptococcus pyogenes will not allow Streptococcus pyogenes to be coated with C3b, and now makes it less delicious for phagocytes to eat. What about viral infections? So let's take a look at this cartoon. So these guys are uninfected cells. This guy in the middle is infected. Here it's got the viral genome inside. And then the virus can make a protein. And in this case, in this cartoon, it's a viral fusion protein. So virus needs to only get into one cell here, and then it can fuse with all these other cells and make this giant thing called a syncytium. And now it's got all that protein making machinery. So here's the virus. Little guy in here, it can make a whole bunch of its viral proteins in this syncytium. So adjacent host cells fuse during some viral infections. It's called the syncytium. Viruses can evade the immune system by expressing fake MHC1 molecules. So if the virus makes an MHC1 molecule and a immune system cell comes around and is feeling out the virus and goes, oh, there's an MHC1 molecule. It's a self MHC1. This must be a self cell. I won't destroy you. Way to go, viruses. Yeah, so cytotoxic T lymphocytes can only destroy virally infected cells if they recognize MHC1. And if viruses make a fake MHC1, then they don't get destroyed. So some viral diseases of the skin, chickenpox and shingles. So here's a little guy with chickenpox, and here is a characteristic shingles rash. These are latent infections, so they can stay in your body for a long time. Some bacterial diseases of the upper respiratory system. So Streptococcus pyogenes is the major cause of strep throat. So here's some strep throat. Oh, it's painful. Also called pharyngitis. Strep throat down the road can lead to scarlet fever, rheumatic fever, and glomerulonephritis. Streptococcus pyogenes has numerous virulence factors. So the host, your cells, your phagocytes, can't phagocytize Streptococcus pyogenes.